Here's the very dramatic start. Oh, yes. So today we'll be discussing Rose Rosette. Welcome, everyone, to another session of what are we again? We're the Chat with Green Aggies. And, every single Thursday at 12:12, we discuss a different topic that's particularly relevant for the green industry, specifically of Texas, but sometimes it can relate to other growers as well. And we often have some featured guests that will come and help kind of talk on some of these topics. So today, we have three kind of main topics that we're hitting on. So all about Rose Rosette, and that'll be spoken by Dr. Kevin Ong. We'll be talking about that. Mr. Paul Winsky will be talking about earth-kind rose trials. And lastly, about other rose issues and symptoms you may notice. That'll be by Laura Miller. And Kevin, do you want to take the reins from here? You should be able to share your screen and give your little bit.
Yep, I'm trying to take over control from you. And let's go. Easy. And hopefully, it's running a little slow. All right, can you see that now? All right, folks. Thanks for showing up. Mong reminded me that I was supposed to talk about Rose Rosette. I don't remember when, but this week has been a blur with some of the things in the news. But in terms of Rose Rosette, we use this time to share with the green industry some of the things that are going on. So rather than show you pictures of symptoms and what and tell you about what they are, I'm going to want to share with you some of the research that has been done on Rose Rosette, particularly with the Combating Rose Rosette Disease grant that ended in 2019. Now, I'm trying to figure out why my slides are not moving. Okay, now it is. So a couple of things I do want to point out about this particular project. It ran from about 2014 to 2019. We had over 15 investigators, both in research and extension, working on it from a bunch of different states, from Tennessee, all in Arkansas, all the way up north to Minnesota, and folks in the east in Florida, and folks in the west in California. And the main goal of this project, when it was written, was to look for resistance to Rose Rosette disease. So this was primarily a sort of a breeding proposal. Let's look for resistance so breeders can use it to breed for resistant Rose Rosette resistant plants. So when we talk about the process going about to look for those resistant plants, we tried to take the easy route. We said, we got a whole bunch of cultivars out there. What's out there? Surely there could be one or two or more than two that is resistant to Rose Rosette. But along the same lines too, we had to answer the question, how can we better detect Rose Rosette? Because the symptomology, even today, is varied at best and dependent on, well, not really dependent on the cultivar. We still don't have a full grasp of why some symptoms show up and why others don't. So that's still an unknown. But we are learning more. And of course, the third thing is, we want to understand the disease better. And when we say the disease better, we need to understand the virus that causes the disease. We need to understand the vector that is helping move the virus around. All those things that we had some historical literature to go on with. But, about 3 years in, we realized one thing. A lot of the original research literature, many of it was done with multiflora rose and does not always fit in when we start talking about cultivated roses. So it was kind of a new learning curve for us. Now, so real quickly going through what was done. So we actually had a big variety trial that was out in Tennessee and out in Delaware. So this were run by two different pathologists out there. One is Dr. Mark Wyndham, that's in Tennessee, and then Dr. Tom Evans in Delaware. Each one of those places ran somewhere about 800 or so. And I think Delaware had about over 500 cultivars. So collectively, we're looking at around 1,200. The short answer to the question, is there resistance to Rosette? The answer is no. So we do see symptoms showing up at one point or another, which is suspicious of Rosette. And to some degree, many of those were tested molecularly. And if it does have symptoms, it is positive for Rosette. Now, what information we did manage to glean out of that was the fact that there appears to be what we call tolerance. So some plants seems to get it a lot quicker, and others seems to stay cleaner, better. And yet there's some plants that would test positive, but don't show obvious symptoms. So you got a range there, but nothing which we would truly call immune or truly resistant. We did have some variety trials in Oklahoma as well as in Texas. And in Texas, that was the work of my graduate student. One of the things we did in Texas trial was the monitoring, and well, not just in Texas, but out of Texas A&M, was the monitoring and citizen science effort. So first part is the field trial. How do we put something out there, but then engage the public to help us get data? And so that was what my graduate student got to do, which is a small variety trial. But then she also had to do an extension program where she educate volunteers how to take data. And the trade-off to that is they got a better understanding of what the disease is, what causes it, how it might spread. And so this is one way for us to combat misinformation. The other thing that we try to do with this engagement of the public is the fact that, you know, we are trying a number of different things. So all the investigators are trying different things, and then there are preliminary information that are out there. And our goal was to make sure that people realize, at least the one that we got to touch, that experimental information are often preliminary information. We got a good result at one test. It doesn't mean it's going to work every time. We still have to do some due diligence to get those replications in, to be able to statistically say, yes or no, this is a good management practice. And even then, we were also trying to educate folks, it is not 100% foolproof. So, you know, in other words, there's no silver bullet for this particular disease. The other approach was, how do we get the information, or trying to glean information for resistance? We used rosette.org, that website. One of the things is we use it as a clearinghouse for the information from the research projects. I think that was, we learned very quickly when we just put it out there without disclaimers. People took that and they took that as gospel truth. So we had to do a little bit of damage control after we put that up with disclaimers saying that these are preliminary results. But also on this website, we actually had a reporting tool. And the idea was we wanted to monitor the range, but at the same time, we wanted to capture any information that we could get of people, particularly rose enthusiasts, who knows what kind of varieties of roses they have. And if we have that information, it's sort of like an elimination process. If we know that this variety has symptoms and we test it and it tests positive, well, that is not a candidate for resistance. So we did that, and we did manage to harvest quite a bit of data off that. This is what the splash page of the data website looks like. I just want to show you some cool data that we can get from that website, and you can get this too. On the distribution map on the website, it gives you a picture of where Rosette has been reported and verified. So every one of those green spot lights up a county. And this green spot includes data that had been in literature, and also reports that have come in through the Rosette portal, and somebody looked at it and said, yeah, it looks like Rosette. We're going to verify it, Rosette. Now, I also want to explain to you that some of those cases that we get through the Rosette portal, we do ask for a sample to test for it. Now, this is what you see on the website. Now, what you do not see on the website is a map like this, that on the back end, we're able to split it down even further to say, how was those verification done? So if you look at the different color schemes that we have there, unknown is probably from a historical literature, which basically says, we saw symptoms of Rosette. You know, it's in literature, it's published, so we stick it up there. Expertise is in literature that somebody said that they identified it, and we know this person is legitimate, so we put it as expertise. If it came in and was identified on a photo, it will be classified as verified with a photo. If it was verified by a specimen, in other words, somebody got a sample in and looked at it and made a call. So that was physical touch of the specimen. And then the darkest one is when we know we actually had the specimen and we ran a genetic test on it. So in some sense, this data for the researchers gives us an idea of where we can put our confidence in the information. So this is just kind of to let you guys know that, you know, what you see outside, we try and summarize things, but on the back end, there's a lot more layers that we're looking at. So I like to show this map because it's one of those that we changed the colors a little bit. The counties that are shaded in yellow are basically what had been reported in literature. So many of this literature was done before 2011. And 2011 is an interesting year because that's the year that we actually had our first genomic tool. That was the year that we had the official scientific report that indicated that Rosette disease was caused by the Rosette virus. The blue dots, or the blue shaded areas, are information that came in through the Rosette portal. So this was actually launched, I believe, in 2015. So late 2015. So, and where you see green dots, that's where is an overlap, where we actually have a report in through the portal, but it was also a county that previously had a literature report at. The basic point I want to point out here for you to look at is, if you look at the yellow, you would say that would be all the information and literature up to 2002. And then beyond that, what you see is you can make the assumption that the range of Rosette is expanding. Now, is it truly expanding? We don't know, but what we can say is when we look for it, it's there. Those are the blue dots and the green dots, and it looks like it's going further out than where it was originally reported and summarized in 2002. So the biology of it, and this is sort of a refresher for most of you. Remember, when you talk about Rosette disease, there are three things involved. Number one is the pathogen. So the pathogen is the thing that causes the disease. The pathogen is a virus. It's called Rosette virus. Rosette virus causes Rosette disease. Rosette virus needs an Uber driver to get to the rose. And that Uber driver is called the vector, which is an aerophyte mite. In this case, it's called Phyllocops fructiferus. Now, so far, and in literature, we Phyllocoptes fructiferus is the only aerophyte mite that has been identified as the vector. There are about 8 different aerophyte mites. These are microscopic mites that can be found on roses. Now, interestingly, in Texas, for the most part, is Phyllocoptes fructiferus, but we do know that in the McKinney area, Dr. Mark Winden had done some survey there. He did find at least 2 other species of aerophyte mites that are present. So keep that in mind. There is some work being done right now, waiting for more funding to look and see if some of this other aerophyte mites could be potential vectors of Rosette virus. We don't have an answer to that. So the story that is true and scientifically tested right now is the fact that aerophyte mite Phyllocoptes fructiferus is the vector. Let's see. So the vector carries the virus to a healthy plant and infects it. Thing to remember here, the virus does not move on its own. It does not have legs. It does not have a means to fly, but it relies primarily on the mite. Now, there are several other ways that the virus can get from plant to plant. One of it is by grafting. This has been shown in literature. You can take a bud from an infected plant, put it on a new plant, and that new plant will get infected. But what has not been reported in literature, and my student is actually working on a publication on this was part of her research, is that this virus appears to be able to move from plant to plant through root grafting. This is not uncommon. There are other rose viruses. So Rosette virus is not the only rose virus. There are several other viruses that infect roses, and at least on the rose mosaic virus complex, it's been shown that they can move from an infected plant to a healthy plant through grafted roots. So that is not, a surprise to us. So the next few slides is just showing you great, you could say symptomatic pictures of the different symptoms. And I just want to make one comment about that. When you look at different cultivars of roses, the symptomology ranges quite a bit. About 10 years ago, I actually had an undergraduate student try to catalog those symptoms, and it was all over the board. And I told her, okay, we're just going to catalog those symptoms on knockouts. Should be simple enough. One variety. And this was the original, the red knockouts, and then we can see what the pattern is. And guess what? No patterns. So what's going on here? So there are a few theories here. Symptom expression could be tied in with some of the environmental factors. Symptom expression could be tied in with co-infection with other viruses or effects with other type of disease problems on the plant. All those are right now theories, and some of it are being tested. But the most common symptom that you are going to find with rosette on cultivated roses are witch's broom. Something similar to what you see on the left, which looks like a little pom-pom at the end of a branch. And very often with the shrub roses, you're also going to see thickened canes, additional or extra thorniness, and sometimes, not always, sometimes the reddish coloration. So if you see witch's broom and you see a lot of thorns on a cultivar that's not supposed to be thorny, but it's not red, it could still be rosette. Just remember it this way. With some of you guys that do the landscape stuff, have asked a lot of questions about, what can you do? I mean, you all say control the mites. Is there really any good stuff out there to use as a miticide? Well, I'm gonna share with you some work. This is a preliminary work, for out of Tennessee, and Mark has been working. I think they have 2 additional year of data right now, that they are compiling. So I'm just sharing with you some of the work that they did about 2 years ago. So 2 trials, 2 years. Now they were just having fun with the products. Okay, so they applied these products weekly at the highest rate, no rotation, every week, dump it on those plants. Their goal was, does any one of this miticide works? And surprise, because his theory was none of them would work. But surprise, surprise, some seems to be able to control rosette disease over a period of 2 years. Now, I do want to explain one thing again. This was products applied weekly at the highest rate throughout the season. This is not feasible. This is not according to label. Do not do this. This is just for your information to help you understand the logic of what researchers think and go through as they refine some of that project. So what are the products that he found to be effective against the um, uh, to reduce rosette incidence? Now, if you notice, what I say was reduce incidence or suppress incidence of rosette disease. Did he do anything to look at the mite or mite population? The answer to that is no, he did not. So there's still a gap in the information. All he did know with this test was, I dump this stuff and I don't get rosette disease symptoms. So keep in mind, there's still a gap. But what it does is it gives us an idea. Well, these are the 4 products that are out there that he used on a weekly basis, not according to label, that seem to show that plants that didn't result in rosette disease symptoms. So this is now taken to a little bit better trial where rates are being looked at, where intervals of 2 weeks to 4 weeks are also being looked at. So, this, I would say, stay tuned for the data. Other products in the trial includes carbaryl, some horticultural oils, and abamectin. So early on, I think we put it out there that folks can use abamectin. Well, in this trial, abamectin was kind of useless. Dr. Wyndham's words, not mine. Now, in terms of residential landscapes, particularly homeowners that has been bugging extension folks, what do we do? What can we use? Well, horticultural oils that was put out there, he tried a few of those summer oils, I think. It was not really effective when mite pressure is high. And I asked him, what do you mean by mite pressure is high? When it appears that there is a large populations of mite in the environment. Well, how do you know that? Well, we just capture the mites that's in the air in the garden. Okay, doesn't mean it might land on those plants. Well, that's true. But nonetheless, his theory on the horticultural oils is it basically creates an environment that's, that the mice does not like. Okay. And the other product that was tested was the bifenthrin, and this is what he suggests to folks, homeowners, to use if they want to use anything at all. If you use it properly, it's not super effective, but he feels that it's still a good thing to use as a preventative in a home garden type situation. But I told him, I said, I would be more comfortable when I see more data. So that's just to share with you a little bit of what some of those things are that we have done so far. Anyway.
If I may
Yep.
say something.
Go ahead.
Representing the industry, a bifenthrin application also has some secondary effects on things, on non-target organisms. So that should also be considered in their selection for alternatives.
Yeah, and so when we have those researchers get together, we always have a little bit of an argument with what to put out there and what not to put. I know Dr. Wyndham actually has 2 more additional years of data that is evaluating right now. Um, the last time I spoke with him about the mitosite stuff, he wasn't very keen, I guess you could say, with the data. I think Laura, you might be aware of some of those too. I don't think he was keen with what he got.
No, I don't really recommend, though I do have people that do use miticides to try to limit the disease spread. I can't really feel good about making a recommendation that that's good management.
And I think that's a kind of consensus right now. There's some miticides out there, but um, it doesn't give as good of a, a result that I think many of us would like to have. I know I'm running out of time, but I want to share with you one other thing that we do here with roses. So one of the projects that we have here in my lab is we are part of the National Clean Plant Network. So what we do here is we help some of the collections to test them for a bunch of viruses, not only Rosette, but 5 other viruses, and so that they could produce what we call virus tested, and some people might use the word, I use quotes, clean plant, clear from viruses. So we are working with the Florida Southern College Rose Collection, which is Old Garden Roses. And we're also working with the Antique Rose Emporium to help them establish a clean plant collection there. The idea there is if you start clean, hopefully, you have better success in growing nice roses. We're also working with Dr. Dave Burns here in his breeding program, providing this sort of screening service so that with the breeding stocks and all that is to start clean really early. Because I think, I don't think, one of the things that you need to understand is if you start off with, let's say, a plant that is infected by some kind of viruses, and you are using it in crosses, there's that possibility that that virus will be continually transferred down to the progeny. So in this type of situation, we want to make sure that we start with a clean or virus tested material very early on. Now, in terms of, and this is part for the plant diagnostic folks, people that enjoy diseases like myself. When we do virus tests, the question always comes out, how do you know that test actually works? How do you know that when you get a positive, it's really a positive and you didn't do something or you didn't sneeze into the tube? Well, you need a positive control. So we actually have a little project where one of my colleagues in Beltsville is the curator of our rose virus collection. So yes, in Beltsville, Maryland, there's a greenhouse that's secure that has about 60 rose plants, and every one of those rose plants have some kind of virus in it that we can go to and get so that we know and can test for, and we call that our positive control. It's pretty cool stuff. So what you might call that's the maximum security facility for rose viruses so that we can utilize them to ensure that our test actually works. So that's kind of like to ensure our ability to not only start, but also to stay clean. That's kind of what I wanted to share with you guys today. Before we got started, I said I was real busy this last few days dealing with some news and guidance to our county agents around the state regarding to people receiving packages in the mail that contain seed that they did not order. So if you haven't heard that story, a bunch of people around the country got packages that has labels appearing postmarked from China. And when they open the packet up, there are seed packages in there. And it's all different kinds of seeds. And so what happened was we were given the instruction to pass the word out. if people get those kind of packages, they're supposed to hang on to them, contact USDA. Do not plant them. Don't toss them away, because there was we didn't know what kind of seeds there were. We didn't want to run the risk that it might be a noxious weed that could be detrimental to our environment and our economy and our natural resources here. Or worse still, it was a seed that maybe harbor a pathogen. So that was the big thing in the news. So if you get one of these packages, or if you have a customer that comes in and says, hey, I got a packet of seeds that I didn't order, and asks you, this is what you should do. Tell them to call USDA or call Texas Department of Ag, let them know. And there's also an email address for them. If they can take a picture of the label and of the seeds, somebody from USDA will contact them and come and get those seeds for evaluation. What you should not do, you should not go, ooh, that's cool, new seeds, go plant them and see what it is. Please do not do that. And also don't toss it away, because we don't want the possibility of it getting out of those bags and then germinating at the landfill area and start taking away, if it's noxious, it has the potential to take over part of that area. So I'm going to stop right here and stop sharing. Back to you, Irfan.
Yeah, absolutely. We've got a quick question from Kathleen Ramey, who's saying that there are many roses in Virginia. Rose Rosette appeared. The Extension Office advised to pull them out and destroy them to prevent spreading the disease. So I've been reluctant to add roses to my garden in Texas. How common is this disease in Texas?
So that... Go ahead, Laura, if you want to answer. Okay, so if you want to look and see where Rose Rosette is in Texas, go to the roserosette.org website and distribution map. You can actually zoom in on Texas and see where we have had reports. Some areas are what I would call have higher disease pressure than others. So if you talk about the Dallas-Fort Worth Metroplex area, I would call that a high disease pressure area. We do have places right now in Williamson County and Travis County. This is Austin area. I would still say at this time, that area is still a low pressure area. So what does that mean? The recommendation that we have still is to say, if you get symptoms, pull out the plant, get rid of it. And when we say pull out the plant, pull it, bag it. The one thing you want to avoid is to give an opportunity for those mites to jump off and move to another plant and perhaps carrying the virus with it. So if you can bag it, cut it, and then basically have it disposed appropriately, landfill is fine in this case. Now, if you're in a high disease pressure, folks said, can I still plant roses? I said, absolutely. But then what should you do about it? Now, in a high disease pressure area, I've been conflicted because, if you pull out a plant and you put a new plant, that's a very good likelihood that you might see symptoms within a year or two. And so sometimes I've told folks that said, I have had this plant for 10 years and it has only one branch that shows symptoms. I said, prune off those branches, then just enjoy it. I want you guys to realize this, that suggestion that I just provided, pruning off the symptoms, is just a cosmetic fix. That plant is still infected. That plant still has the virus. A mite could come to that plant and still get that virus. But I figured in a high disease pressure area, you know what? Enjoy the plant. In other words, I went into a hospice mode.
Thank you, Kevin. All righty. Shall we move on to just to stay a little timely here? We'll move on to Paul, and then we can take some more questions at the end if we got it.
Here's the thing. Never believe Dr. Kevin Ong when he said, oh, I only need 20 minutes, and then 45 minutes passed.
What? It's already 45 minutes?
Yeah, man.
Oh,
And why didn't somebody stop me?
30 minutes, Kevin.
Well, I messaged you. You got 38 seconds left, like 8 minutes ago. But
Well, Laura,
you
I
didn't see
just
the message.
exaggerated a
Sorry,
little
dude.
bit. I mean, only a little bit. So,
That's all right.
So this is just for the panelists. You know, just be aware. Dr. Ong's time. I mean, his math is,
Well, let's let Paul have his time.
Yeah, let's go ahead.
All righty. So, Irfan, can I drive this or?
You may be able
advance?
to do remote control.
Okay.
See if you can do that. If you scroll to the top, you should potentially have that as an option. I'm not sure if you can as a co-host.
Where would I see that at?
So right now, it should say something about like sharing screen when you scroll to the very top. Some options about muting yourself and stopping your video should be there, and then the remote control would be there. If it's not, I can just kind of drive it for you.
Okay, why don't we do that so we don't waste any more time?
Sure
So,
thing.
all right, so let's talk about Earthkind. This program has been around, I guess, since about the mid to late 90s. Steve George and I believe Dr. Wayne McKay, when he was still with A&M, they were sort of the godfathers of this. And it really, the Earthkind approach is a sustainable approach. Minimal inputs into the landscape, and really finding those plants that respond to this type of approach. So one of the advantages is it does preserve and protect the environment. For the homeowner, they can be successful in using these practices. They are not difficult to do, and overall, it is a sustainable approach. So what we did, next slide, is in working with Dr. George and the Houston Rose Society, they wanted to look at, you can go back one, I'm sorry, if you don't mind. They wanted to look at what they call the modern rose trial. Everyone knows, especially down here in the Houston area, growing hybrid teas can be quite difficult. And with the help of Gay Hammond from the Rose Society, she felt that some of the Cordes roses, which are bred in Germany, and they have no, when they are screened early through, there's no fungicides, pesticides, they minimal inputs as they're screening. She felt that some of those varieties would do well down here in Texas. So we had these 20 varieties that we, this list that we developed. Carefree Beauty, as Dr. Ong said, we had our positive control in there. Carefree Beauty was there because it's already a Texas superstar that performs well. And then on recommendation, we put Shreveport in, which is a black spot magnet. So we wanted to make sure that if black spot was in the area, we would find it. And so if we go to the next slide, we can see what some of these what the roses, the colors look like. So Beverly was one that did extremely well for us. Pink Enchantment. So they have that hybrid tea look, and they have fragrance, which is good because we're missing that with a lot of the current varieties that we have down here. Plum Perfect was one that did extremely well for us. So this gives you an idea of what the potential was as we were going into this. Next slide. So where were we? The trial was planted in 14, 15, oh, back one, please. This was an urban setting. This was on the corner of Old Spanish Trail and Fannin. We partnered with MD Anderson, and the trial was in long enough that we made it in Google Earth. So I feel pretty proud about that. But you can see where the trial was located. If you look at that second bed in, there was a concrete slab that went about 3/4 of the way down that bed and was probably about 18 inches. It was about 18 inches deep. So it was not very good soil, which was fine because one of the first steps you do when you want to incorporate using Earthkind practices is we always recommend, A, we took a soil test, and then B, we amend the beds with 3 inches of compost, and we did do that. And so as the trial went on, and this was over 3 years, the plants thrived. Of course, we had some losers, which you would expect. You don't expect all 20 to be winners. So next slide. So this was early on in the trial, and you can see the, how the location was. These beds were about 100 feet long. I think the roses were on roughly 8-foot centers, about 10 feet wide. And we had 4 replications, so 20 roses in a row and replicated 4 times. We had 2 teams of master gardeners and volunteers from the Houston Rose Society that would collect data on a monthly basis starting in March and going through September for 3 years. Next slide. So this is just really the collaboration, which I thought was really a good part of it. I was relatively new to Extension. I've done a lot of trial research work, new product development, but this was really fun working with MD Anderson, who felt that, this is sustainable approach to this growing was something they were interested. And the folks at both the master gardeners are always wanting to get in to do trials and to work and to learn new things. And the Houston Rose Society, a lot of those people, they, just their background in growing roses and getting them planted and the little tricks I learned in working with those folks. So it was a real interesting collaboration. It worked out really well. And I think we're all happy with how it finished up. So next slide. So I was really impressed. MD Anderson, they really wanted to bring some publicity to it. So they had this beautiful sign done for us. So as their employees would be walking, either during lunch or going passing between buildings, they would stop and check this out. If we were out there working or collecting data or taking pictures, they would stop and ask us about questions. And the other thing I thought was really helpful was, in that lower right-hand corner there, all those companies, Rainbird and New Flora and Nature's Way, all the materials that we use for this Chambly's Rose Nursery, it was all donated material in order to get this up and running. So, not only working with the volunteer groups, but the industry folks that donated product to us in order to help us get this trial in place. Next slide. So this is just a copy of one of the data sheets. We looked at everything from number of blooms, percentage of the plant that was covered in blooms. It then received an overall quality. And then you can see across the top, drought tolerance, leaf blackening, black spot, powdery mildew, alternaria petal blight, rosette, general insect or mite tolerance, and then chili thrips. So as Kevin had said about training these folks, master gardeners, and even the rosarians, having them look at this a little bit differently from a research perspective, as opposed to just the beauty of the rose and the aesthetics of it. So this was just an idea of the information that was collected. Next slide. So this is what I'm going to do is just really touch on some of the highlights, the plants that performed extremely well. We're still crunching the numbers, and the other thing I want to do is we've got a little bit of missing data, so we're trying to figure out how to handle that. But this same trial was replicated with Dr. David Zlezak up at University of Wisconsin-Whitewater. So this same trial was put in at the Green Bay Botanic Garden. So we've got data from zone 4 and we've got data from zone 9. So I'm really curious to see across that broad environmental growing conditions, which roses really rose to the top, and are they the same ones, or are we going to see southern differences and northern differences? But Pink Enchantment had these very full flowers, very good flower production over the year. over the growing season, and a really good fragrance, which was really nice. Not overpowering, but when you were near it, you knew it was a rose, and you knew it was, it had a really good fragrance. Very little black spot, very clean. These plants were never sprayed. And we could see as I would go through and check on these, I saw a lot of long-legged flies. We would see some of the other beneficial insects that were in the area, but we never had any aphid problems. We never had, just none of the issues that we would see maybe under conventional methods. So these plants are really healthy and perform really well. And you can see there's some in the background there that just didn't make it, and that's going to be expected. Next slide. So this is Gay Hammond. When we received it, it still didn't have its name yet. It was Zlezak #7. So this was bred by Dr. David Zlezak. But this plant, I was really impressed. Now, this one doesn't have a fragrance, but as a shrub rose, this thing was a beast. Very good foliage throughout the year. Nice dark green foliage. You could see the flower production on it. In the summer, yellows down here will fade on us, so it would open. We'd have a good yellow opening, the first day, but by later in that day, that color does fade. But in the spring and fall, that yellow color holds much better. And the next slide is sort of a nice image where this is rep 2, 3, and 4, and the 3.
Sorry.
No problem. And the 3 flowers, 3 roses that look like they're white, they're actually at a younger stage of Gay Hammond. And you can see the uniformity in flower production and growth habit. So this is one as a shrub rose. And if you're looking for a yellow for Texas, which I'm sure a lot of people want that yellow rose of Texas, this is one that you really might want to consider if you're on the production side, or even if you're on the landscape side. This is very, very little input, and this plant is going to thrive for you. And the last rose I want to look at, next slide, please. This is Plum Perfect, and you can see the flower production on this. And these roses were never pruned. So over the course of the 3.5, 4 years, we just let them grow. Nothing was pruned, so we didn't go out. The rule of thumb down here is, come Valentine's Day, you prune your roses back. These plants were never pruned. They just continually grew, and you can see the flower production on that. It's got that hybrid tea look, and this is another one that has really good fragrance. So both the Plum Perfect and the first one, they're both Cordis roses. I would say there's probably 2, maybe 3 other varieties that could potentially be slated as Earthkind winners. But it was overall, the trial was a lot of fun. Weed populations were a challenge, of course, but the master gardeners and the folks from the Houston Rose Society and our contacts at MD Anderson were more than willing to pitch in and help as needed. So that's the update from the trial that we did down here in Houston.
Thank you so much, Paul. Sorry for the slides appear to sometimes like to jump 2 at a time. So
not a problem.
Jump back, so it's acting a bit funny. For those of you that have not yet filled out the poll, there's a poll with like 3 questions. If you don't mind answering those at your earliest convenience, especially before you jump off, that'd be greatly appreciated. So last, we're going into Laura Miller talking about other rose symptoms.
Yeah, actually, I'm going to talk about my trials. So
and your trials.
But go ahead. Let's throw it out there.
She's going to talk about things.
The first slide is all about other rose symptoms. So take a look at this, folks. The picture on the left came to me in email last week. The picture in the middle I took in the Will Rogers compound a couple of years ago. And the picture in the middle, and then the picture on the right, I took at Dr. David Burns' rose trials that he had at Seville Farms here in Tarrant County a couple of years ago. So which one of those do you all think is rose rosette disease? Because one of them is. Lab confirmed.
You sure just one of them?
Mm-hmm. I'm sure.
The middle one?
Yeah, Irfan, good job. The middle one, and you can see the witch's broom. You can see how part of the plant just looks so different from the rest of the plant. You can see like the healthy plant and then the infected looking part, the symptomatic part. You can see the red coloration. You can see the excess thorniness. You can pretty much see all the rose rosette symptoms there, and that's a drift rose. And there were, I don't know, about a half a mile of those, and they're all gone now. Now that one, the other two, what do y'all think is going on? It's something that happens every day, in my opinion.
Oh, it's
Herbicide
that.
Some
drift?
herbicide.
Herbicide?
It's herbicide, yeah. In both of those situations, the plants were, thank you, Kevin Ong. In both of those other two situations, those plants were both sprayed with Roundup. And so we know now the one that I got in the email, that could have been rose rosette possibly, but I didn't know that it wasn't. But the one on the far right from Dr. Burns' trials, that was before rose rosette was in that part of the county and it hadn't been identified there. So we know it was Roundup and Roundup had been applied. But that one really, I don't see other symptoms other than just the distorted leaves, the small, narrow, distorted leaves, which does happen all the live long day. Okay, so let's go on and talk about a couple of other things going on around here in the hot spot of Dallas-Fort Worth. This is the Fort Worth Botanic Garden. This is a picture looking down the rose ramp in the old historic part of the garden. At one time, it was completely 100% covered with knockout roses, that ramp was. In 2012, they started having symptoms of rose rosette disease. And it was really frustrating for Dr. Ong because we would send them in and then they would come back negative, though the symptoms were there, the mites were there, everything pointed toward rose rosette disease. And we're reasonably confident it was because it kept spreading. And finally in 2015, they decided to just take out all the roses in the historic rose garden in the Fort Worth Botanic Garden. And if you looked at a picture of that in the winter, early spring of 2016, that would have been the pansy garden of Fort Worth Botanic Garden. But in 2017, they decided to replant. Following some of the recommendations from Dr. Wyndham up in Tennessee, they decided to spread the roses out a lot more than they had been so that the mites couldn't just, walk from or wiggle from rose to rose. And also to plant other plants in between the roses. So you can see there's a lot more other plant material there. And they took out 2,500 plus roses and planted 289 roses. So you can see that there are many, many fewer roses in the rose garden, but it's still a rose garden. The last update I've got, they've removed 19 plants. And if you'll go to that next slide, you can see the very first hole in the rose garden there. You can see in the middle of all the beautiful Cora vinca, there is an empty spot, and that was a fire color opal, just like the one in the background. That was the very first one to get removed. And that's been their strategies thus far. They've, of course, got the barriers of other plants. They've got the plants spaced out, and they're regularly scouting for symptoms and removing. And from time to time, they have used miticides in the middle of the summer to try to stop the spread of rose rosette from the mites. But that's kind of their strategy. It's working reasonably well, not as well as I had hoped, but it is working reasonably well. And now, I need to talk about the City of Grapevine trials. I need to give a big shout out to Robbie from the Antique Rose Emporium because she's helped me so much with choosing cultivars for these trials. And a little history on this. The original planting, and you can kind of see down there in the picture what the area looks like. It's a trailhead in the city of Grapevine, and the city of Grapevine does maintain it, or their contractor TerraCare maintains it by remulching. You know, there is drip irrigation there, so it's not straight up a no care trial, but it is no chemical application. It was originally planted sometime back when Dr. Steve George started doing earth-kind rose trials, and in 2015, all of those roses, except for one cultivar in that original trial, were showing symptoms, and they were enormous plants that had to be pulled out with a truck and chains, and you know, it was a big mess. And then in June 2016 was when we got around to replanting that, and I'm going to show you lists of all the plants. In November 2017, oops, go back. In November 2017, we removed all except for 4 cultivars, and one of those was actually one of the cultivars that did not show any symptoms from the very first trial. And then in December 2017, we replanted everything but those 4. And those are all still there. We haven't seen symptoms since. As Paul mentioned, this is a great partnership. There are volunteers who are Tarrant County Master Gardeners, but also Grapevine Garden Club members who monthly have been collecting data on rose rosette disease symptoms, black spot symptoms, cercosporal leaf spot we've also seen, listing insects, and just kind of keeping an eye on everything. So let's go on to the cultivars and stuff. When we removed it, and this is important to show kind of how hard removal is, it's really difficult to try to get all the roots out from a rose. We worked really hard at this, and we also had these nice barrels with bags to try to get all the roots and bag to contain the mites, and it was a lot of work. I would just say that that's, you know, that's our recommendation, but it's a difficult thing to do. So go ahead. All right, so this is the original earth-kind rose trial that was in this location. There were 20 cultivars, and they were replicated 3 times down those long winding beds. And these are all the things that developed rose rosette symptoms, all the things except for one. And that one, let's see if we can figure it out as we go on. Go ahead and click over there.
Laughter?
Yeah, it's laughter. Probably heard me say this, but there it is. And that's actually a picture of laughter. We planted some extra ones down at the end around the trail sign. It's that larger kind of coral colored rose in the background. So that's laughter, and it was replanted in 2015 along with all of these other cultivars in 4 replications. So we went from 20 cultivars and 3 replications to 16 cultivars, 4 replications. Now 2017, this was when we replanted. We left Laughter, Caldwell Pink, which never showed symptoms, Love, which never showed symptoms, and Basie's Purple, which never showed symptoms. Basie's Purple is a rugosa type, so we did add some more rugosa types. Robbie really helped me with this list. We've got a lot of cool things. You'll see we have Plum Perfect, just like in Paul's trial, which has been a very pretty rose. And we also have some miniatures, and those were from Dr. Burns' Ralph Moore collection. One of Ralph Moore's roses often shows up on lists of roses that haven't really shown symptoms, and that's Little Buckaroo. You'll see Little Buckaroo is not here, but these are other miniatures. So far, all of these have not shown, this is a picture in April of this year, 2020, have not shown rose rosette disease symptoms. So there they are. Also in 2019, the city of Keller, at the Keller Library, we put in a smaller trial, 6 cultivars, 3 reps. This is a picture of Lady Pamela Carroll, also from Antique Rose Emporium for the most part. And we've got that little buckaroo in there. And these have not shown any symptoms of rose rosette disease. So,
So I think that concluded all your slides, right?
that concluded all my slides. I was like, dang, I don't remember that.
What is this?
All right.
So this is just somewhat kind of related to looking at some symptoms on the roses. This was just kind of like to tag on where you might see some things that may look like witch's broom, or they might look like rose rosette. But I think, like you had shown, it was the combination of all the symptoms and then getting lab confirmed that can kind of make us really certain. And in this case, we're seeing symptoms that may kind of mimic rose rosette in some ways. We look a little bit closer. Again, we can see like some crinkly leaves as well versus some normal growth. You can get some discoloration, that kind of reddening that Dr. Ong mentioned, and that we also saw in the rose rosette on some of those stems. And this, in this case, is symptomology of chili thrips. So chili thrips are very small, especially compared to flower thrips, and they can cause symptoms that can be akin to, at least in some ways, to rose rosette. So you can get some kind of witch's broom and some discoloration. So that's something also just to be aware of. I think that officially concludes kind of our presentation format. There was a question from one of the audience members. Is there any info on the roses in the Houston trial regarding rose rosette disease?
We did not, when the trial was in, we did not see any major breakouts or any symptoms of rose rosette. And that trial was in for, like I said, somewhere between 3.5, 4 years. You know, unfortunately, that area is now a parking lot because, like anything else, when MD Anderson gets some money, they're putting a building in. So they are expanding the building next door. And so we were able to finish the trial, get the data. They lifted some of the roses and used them around in their landscape. But we didn't see any in that trial.
I have a question for Laura. Well, you know, Paul, your plants has already been destroyed. So for Laura, although, I mean, you know, you did the replanting at the end of 2017 and then some more planting in 2019. But the thing is, it may take some time for from the first mite infestation to the symptoms showing up. So we can't say at this point that all those cultivars are resistant.
Nope, can't say that. And it's taken as long as 18 months documented in literature from purposeful inoculation with infected mites to symptoms. So we know it can take a long time. It's interesting to me, though, the pressure's definitely less because that 2015 trial was almost completely infected in, you know, a year and a half, versus now we've gone quite a while and they still look fine. So it's interesting. Pressure's less.
Yeah, the pressure is less. And also it could be, well, the pressure is less. It just could be that, you know, whatever the mice the mice haven't got to those roses yet, or the mice haven't you know, visited some infected roses,
right,
and then coming back to these roses, right?
Sure. And there are a lot less roses in the Dallas-Fort Worth Metroplex as a whole. The city of Southlake took out $500,000 worth of roses from the medians in Southlake Boulevard. There are just fewer roses around these days.
So there's a comment in the chat that you know, someone said that I probably won't plant any roses, and then Robbie said, please plant roses. I think moderation, you know, just doing something in moderation is probably the key. Don't overplant, but, you know, one of you that you enjoy, I think that's probably okay.
Yeah, I personally planted roses last year. I did. So
I want to throw in an idea here for you guys. So it has been my contention that, you know, if you treat roses like a 2-year plant, like some gardens too. You're rarely going to run into rosette issues, disease issues, if you treat it like a 2-year plant. But anyway, thanks, guys. I got to run. I got to be on another Zoom in 3 minutes.
Thank you so much, Dr.
Awesome,
Ong.
Dr.
You guys.
Ong.
This is Robbie. I agree with Dr. Ong, and there's no profit in it for me to say plant roses at this stage of the game. But my sister's house is up in the outside of Denton, and we planted probably 10 different cultivars and 60 or 70 plants, and a good many of them got rosette, but not all of them did. So that's not to say that every plant in your landscape's necessarily going to get rosette just because you're in a hot spot. It will depend on, like the Fort Worth Botanic Gardens, how they're interplanted, what the dynamics of the wind are in the house. The roses that are prettiest are up closest to the house where the winds aren't blowing the mites up into them. Thank you.
Thank you. There was I think this is back towards Paul, another question that came in asking about just how prevalent or what's the rosette disease pressure in the Houston area. I suspect this is relating just to, can we really, infer any type of resistance or anything like that based on these field trials? And it would help to know if there's a lot of pressure, then, maybe it suggests it, but if there's not a whole lot of pressure, then, I mean, we don't really know.
Yeah, I would not say for that, we didn't see it. And like you said, this is a 3.5 year, 4-year picture. Could they have gotten it? Absolutely, at some point. But is there pressure? There are pockets in Harris County in Houston where we see it, but it's nowhere to the degree like up where Laura is. And so I don't know. And knockout rose, there are drifts of them, and they're using them in developments and parks there. But it's not to that extent. I know every time Kevin comes down, he seems to always find it as he's driving around to come before he gives his talk. So we do have it, but it's so again, I would not say any of the plants that we trialed that were clean. They're definitely, I would not say they're resistant. I would just say, we didn't see it in the trial from the time that we had it.
And I think,
Sure.
and I think this relates back to, we had a question from Neil Sperry asking whether there are any beginnings of a list that would want to give out to media of resistant varieties. And Dr. Ong gave, I think, a good answer there in that chat box, but basically we don't know yet. It'd be a little too early to provide even a preliminary list at this point.
This is Neil, and I would add a comment for those who say let's plant roses. Those of us who are giving that advice, as well as bona fide nursery professionals, are going to be held accountable for that advice. It needs to be given with an asterisk of, if you want to do so, but please know that they won't last as long as you would like them to. And,
Right.
and we need to warn people.
Right.
It's been 10 years since I have seen in the Dallas-Fort Worth area a beautiful blooming rose plant. That's pressure. It is awful up here. I don't know if Laura has had that same experience, but it's just dreadful.
No, Neil, I mean, I showed you a picture of beautiful blooming ones from April in grapevine. So they looked great.
Yeah, and that's true. And I was doing screen captures of those. That's why I was hoping you would have some lists.
Good.
He's going to look for those in actual grapevine, see if those photos are actually from there or not.
Sure, you can go on Google Earth right now. 501 Shady Lane across the street.
Some of us were not out and about in April. Some of us were
I did want
in our
to say
houses.
something about, Robbie made a comment in the chat about the Gulf Coast not seeing a lot of rosette disease. And there was this theory that it wasn't going to go south of I-20. And they held that theory for a long time, that the mites weren't going to, move south of I-20, and they did mite surveys and a lot of stuff. And I think we in Texas have busted that out. So but there's, I don't know. It's complicated. It's a virus. Viruses are complicated.
I think that warning is a good practice. We sometimes we can't treat things just like carefree. We should be taking measures, paying attention, and knowing that there are some of the risks behind these things. In the case of Rosette, and because you don't know where your inoculation source may be. It could be your next door neighbor who's not as careful as you are. So those are the things. And I agree that monoculture is definitely problematic with any type of plant material. So that cycling, with the rose plants may be a strategy for the short run before resistant materials is developed. And then again, even with the materials developed in the future, there may be more resistance. The mites or the virus may evolve too. So I definitely agree. Monoculture is not the way to go in landscape practices.
I'm trying to speak on this.
Just got a hand up and someone saying a little comment there.
John?
Sure. Can you hear me?
Yes, we can hear you.
Hi, just for your information, I led Dr. George's Earthkind trial in Galveston County in the early 2000s, and there was absolutely no sign of rosette disease back then. And I think we were looking for it based on a research that some guy in southern Florida had done, and he in amongst viruses and all that stuff. And again, that was a long time ago. But I also share with you, I was the one that sent the note to you guys last week, what the pressure we're seeing right now, and I'm talking about south of I-10, if you want to think of the southeast corner of Galveston County, is immense chili pressure. I mean, we got all the situations. I mean, I got a rose bed where I have test roses and newly hybridized roses, and literally the chili thrips are absolutely devastating. I mean, it's nasty. But again, I've also got a rose bed at the Galveston Extension office, Dr. William Johnson's area, and we see absolutely, there's no rosette disease apparent down there, and there's no chili thrips. So maybe the marking line might be Highway 518 at the moment versus I-10. But anyway, I just wanted to share that with you guys.
Thank you.
I would say I can recommend a couple of landscape practices. Just as we mentioned before, spacing the roses farther out apart from each other, interplanting other plants that are not host to the mite, which I don't think there's other hosts other than the Rosaceae. Not using leaf blowers in roses because the mites can be blown around. They do spin little, I got my mite here, my 3D printed guy.
Very nice.
They do spin little threads out of their spinnerets, I guess, and they can blow. So not blowing is a good landscape practice. Trying not to move from an infested area to a clean one when you're working, because the mites can be on your clothes and all of that.
Well, thank y'all so much. I think that concludes our meeting. We've gone on a little bit long today, but thank y'all tuning in for this session, which was on Rose Rosette. Thank you so much to our speakers, Paul Linsky, Laura Miller, and Dr. Kevin Ong, for providing all that highly valuable information for us. And remember, you can always join us every Thursday at 12:12. Their meetings are all on Zoom now, and you should get emails or follow us on Facebook, Chat with Green Aggies Facebook page, to register for further webinars or to see even the recordings. So this recording will also be made available so you can watch. So thank you all for tuning in.